Thank
Okay,
you.
great.
So thanks so much for inviting me here today to present a little bit about myself and the research that I've been doing since I came to UCF. So my name is Elizabeth Brisbois, and I'm an assistant professor in material science and engineering, and also a member of the bionics cluster. So my research lab is down in Lake Nona, really nice campus if you haven't been there. So to start off with, I thought I would just introduce myself a little bit. Previously, before I came to UCF, I was at the University of Michigan, where I did a lot of training both in chemistry, bioengineering, and also in a surgery department at their medical school. And so a lot of my research focuses on biocompatibility of medical devices. We develop different types of biocompatible polymers, especially ones that use nitric oxide chemistry, which I'll talk about some of the details about that in a minute here. But ultimately, we're really interested in using these kind of biocompatible technologies in order to improve the success of a wide range of medical devices. And then in terms of teaching, since I came to UCF, I've been involved in a variety of teaching, both undergraduate and graduate level courses. Everything from MSC's Structure and Property of Materials class, which is kind of introducing undergraduates to the material science engineering field, as well as graduate level courses related to biomaterials and biocompatibility. So that's kind of what I've been involved with teaching-wise. Next, I'll kind of jump into a little bit more about my research area and kind of our motivation and some of our accomplishments kind of highlighted here. So the general motivation for the research that we're doing is the fact that there's a wide range of medical devices that are used in healthcare today, some of which are summarized in this image here. You can see there's a number of different medical devices, everything from small things like different biosensors or catheters, all the way to large devices like hemodialysis or heart-lung machines. And so kind of the overarching goal that I have is to try to study new materials that we could use to fabricate these devices so that they can be more successful when used in patients. And so in terms of like some of our accomplishments so far, I'll talk about this in in the coming slides, but we've been fortunate to secure funding from UCF through the ACER award, as well as through NIH and another organization called the Juvenile Diabetes Research Foundation. I'm also pretty involved with professional societies, specifically the Society for Biomaterials, where I'm currently a secretary treasurer of one of the special interest groups. And then, I've won several different awards over the years, a few that I highlighted here regarding my NIHF 32 fellowship previously and a career development award. And then since coming to UCF, I've been pretty active in trying to build my research group. So currently I have 5 graduate students and postdocs in my group, as well as a number of undergraduates doing research with me. So next, I'll share with you a little bit more about the research area details that we're currently investigating and show you a couple of examples of projects that we're working on. So one of the things with regards to all those medical devices that I showed you on the previous slide is the fact that many of them still suffer from major problems related to blood clots or infection that happens when they're used in patients. And so these clotting and infection problems can cause a lot of issues with regards to the patient's care, but even it can lead to their death and really expensive healthcare costs. So what we want to do is kind of come up with some new ways to design materials that can overcome these clotting and infection issues. And so one of the technologies that we're investigating significantly right now is the ability to use or develop nitric oxide releasing polymers. And nitric oxide is a really important component to these polymers because it has a lot of different biological roles, and in fact it's a molecule that's produced in our body. So as a couple of examples, nitric oxide can kill bacteria. It can prevent platelets from getting activated, which allows us to prevent clotting, and it even has anti-inflammatory properties as well. So the general idea of what we're doing is trying to develop new polymers that have nitric oxide release at similar levels to what is produced in our body. And then we can use those nitric oxide polymers in order to improve the biocompatibility of medical devices. And so we do, really translational research, everything from, synthesizing and developing those new materials to characterizing their biocompatibility both in vitro as well as in various animal models. And then our ultimate goal is, to be able to take some of these technologies, the best ones, and translate them into clinical practice. So next I'll just kind of highlight a couple of the major project areas that we're currently focused on to give you an idea of some of the types of projects we do. So one of the big areas that we're investigating is using these nitric oxide polymers to prevent clotting and infection on different medical devices. And so here I'm just kind of showing you a quick list of some different medical devices where these polymers could be applied. We can apply these polymers to really small devices like insulin infusion cannulas that are part of the insulin pump system, as well as different types of catheters, and even large devices like extracorporeal life support or heart-lung machines, hemodialysis, things like that. And so the pictures that you're seeing on the screen are just kind of demonstrating the significant activity that these nitric oxide release polymers have. So you can kind of see how, in comparison to control surfaces, the nitric oxide release polymers can do a really good job eradicating and killing bacteria, as indicated by all the red bacteria, which shows dead bacteria. And then on the far right, you can see examples of intravascular catheters that we've fabricated that are able to prevent clotting issues. So you can see on the top right is a control intravascular catheter, which is much like what's currently used in patients today and suffers a lot from clotting problems. And then our nitric oxide release catheter that's on the bottom right pretty much has no clotting, which is really nice. Another project area that we're really focused on currently, and we've received funding from the Juvenile Diabetes Research Foundation, is being able to take these nitric oxide polymers and apply them to the insulin pump system that diabetic patients use in order to improve their biocompatibility. And so the major reason this is important is because patients who use the insulin pump, they actually have to place a small catheter tube under their skin, which delivers the insulin from the pump into their body. And this insulin cannula has a lot of problems when it's implanted like that, because we get a lot of inflammation, we get foreign body response, infection issues, and ultimately that can actually encapsulate that cannula and limit the efficient infusion of insulin into their body. And so anyway, this is what we're doing, is we're basically developing a new insulin cannula that has nitric oxide release. And then we're studying it in a rat model to kind of evaluate how well this new polymer system can reduce all those inflammation and encapsulation issues in order to, basically more efficiently infuse insulin into the patient. So in terms of future efforts and some of the opportunities that we're looking for, so obviously we're really interested in applying for a variety of research funding to be able to fund these sort of projects. We've been successful at getting funding from NIH and the JDRF organizations already, but of course we're still pursuing opportunities that are relevant from the National Science Foundation, CDC, and Department of Defense. And then in terms of collaboration, hopefully you could kind of see today that the research we do is pretty multidisciplinary in nature. So we really enjoy working with a variety of experts, everything from chemists and engineers, different people with biological expertise and surgical expertise, and also working with clinicians and hopefully eventually industrial collaborations as well, which will allow us again to hopefully translate some of these technologies into commercial medical device applications. So with that, I'll just quickly wrap up and of course thank the organizers for inviting me today. Thank my research group for all their hard work and efforts. making this research possible, and of course our current funding from NIH and JDRF. And with that, I'll take any questions.